We're covering today macromolecules. As you can see, there's just a nice picture there of some 3D artwork done through computer-generated models of proteins, one of the macromolecules, major macromolecules that we will be focusing on in this lecture. So, quick overview. So, what are the macromolecules? Well, we're going to be talking about sugars or carbohydrates, proteins, and lipids or fats. We're also going to briefly review some chemistry. Mainly, we're going to focus on the chemical bonds, so covalent bonds where electrons are shared, electronegativity, so the affinity for electrons, polar, nonpolar, and of course, we've touched on this briefly back in lecture 1 and your first quiz, which was hydrogen bonds. Hydrogen bonds probably being the more crucial, as hydrogen bonds we will see in various aspects, particularly as we progress into DNA and molecular biology, as well as cellular components and things like that. And then, of course, we'll briefly discuss diets and the breakdown of food for energy. And obviously, that's a hot topic these days. So, obesity, childhood obesity, um, just the whole health and fitness industry, how it's blown up and grown so much. Um, so, we'll touch on things like that. Um, you know, Part of your homework assignment is going to be to talk about diets. So, low carb diets, high protein, high fat. There's all kinds of various diets out there. And so, this is chapter, for example, this particular lecture is going to cover and give you a background into why these types of diets have become so popular, because you have to understand how the macromolecules, the carbohydrates, the proteins, and the fats are utilized in the body at the molecular and cellular level. All right, so let's keep going here. So again, like I said, we're going to touch on it briefly with a chemistry review. So chemistry, what it is. Chemistry explains how elements combine into compounds that make up your body and the bodies of all other living organisms, and how the chemical reactions underlie to function of all cells. So again, compounds, right? We touched on this. Compounds being a substance consisting of two or more um, different elements combined in a fixed ratio. Perfect example, basic chemistry, basic compound, water. Water is H2O, 2 parts hydrogen, 1 part oxygen. There you have basic compound, which is water. Another one that we're going to see, right, is in the carbohydrates, for example, which is glucose, for example, or the blood sugar in your body, that's utilized and broken down for energy use. That is a combination of carbon, hydrogen, and oxygen, also typically in a fixed ratio. Okay, so these are again the more common compounds um, or elements that make up compounds: oxygen, carbon, hydrogen, and nitrogen, which make up 96% of living matter, including us. I want you guys to make sure you understand and review protons, electrons, and neutrons. So remember, protons are positively charged ions, electrons are negatively charged, and neutrons are neutrally charged. They have no charge. Understand what the atomic number is. So the atomic number is a number that's designated to elements based off of the number of protons that they have. And the mass number is the sum of the protons, the positively charged ions, plus the number of neutrons. Other elements of importance, too, just to point out, besides the carbon, hydrogen, oxygen, nitrogen, you should also be familiar with is calcium, phosphorus, potassium, and chlorine as well. So, chemical bonds, okay? So, again, covalent bonds. I want you guys to basically, from these, to understand just the kind of the clear definition. Um, and you should probably, in terms of respect to the exam, be able to identify a particular type of bond, either based off of explanation or a drawing. So covalent bonds, two atoms, each with an unpaired electron in their outer shell, actually share that pair of electrons. And then this ultimately leads to the formation of molecules of different types. Electronegativity is an atom's attraction for shared electrons. So, the more electronegative an atom is, the more strongly it pulls or it attracts electrons to it. So again, if for example in a question or in the readings you come across and you see something where they're talking about a particular atom that has a high electronegativity or high electronegative number, that means that that particular atom wants to attract electrons. Okay, that's like well, let's put it in perspective here, right? It's like a perfect 10. Whether that perfect 10 is the ideal male or ideal female, people are going to be attracted to that perfect 10 person. Same thing here. The higher the electronegativity, the higher the attraction towards that atom. All right, so again, little mnemonic there to kind of help with that. And then nonpolar covalent bonds, electrons that are shared equally between atoms. So again, there's no pushing or pulling of electrons. Electrons aren't bouncing from one atom to another, like they might be for example, in covalent bonds. Here in nonpolar, they're shared equally. Okay. Understands too that there are ranges or numbers associated with these types of bonds. So, for example, nonpolar bonds occur when the difference in electronegativity is between two atoms is less than 0.5. Okay? So again, there's two atoms, and when the difference in electronegativity is less than 0.5, they're going to form a nonpolar covalent bond. So again, electrons are shared equally. Polar bonds occur when the difference in electronegativity between two atoms is between 0.4 and 2.0. And then ionic bonds occur when the difference in electronegativity between two atoms is greater than 2.0. So that's kind of the scale you're working with, is between zero point 0.4 and 2.0 is kind of the electronegativity scale to determine how the electrons are shared between two atoms when they are coming together to form a molecule ultimately. And then an ionic bond is, again, two ions with opposite charges attract to each other. Perfect example is salt, sodium chloride, NaCl. Uh, Sodium is positive, chlorine is negative, opposites attract, ionic bond. And then, of course, a weak but most important type of bond that there is hydrogen bonding. So again, hydrogen bonding we see with water formation, whether it's liquid, gas, or ice. Um, we are going to see hydrogen bonds holding DNA as well together. So your adenines and your thymines and your cytosines linking with your guanines, the building blocks of DNA are held together by hydrogen bonds. Okay, this is then just a basics outline here of what a chemical reaction looks like on paper. Um, so again, just understand that this is what a basic chemical formula looks like. One side you have your reactants. So again, for example, two hydrogens plus one oxygen. This leads to the formation. So this is sort of would be like your equal sign. to your product or the end result, which would be water. Okay? And then just like in math, you have coefficients. So these numbers telling you how much of the hydrogens are coming together plus the oxygen. Okay? So again, reactions make and break chemical bonds. And again, this can be reversed. We could flip the arrow here and we would be showing the breakdown of this water product into its original reactants. So again, chemical reactions can either show reactants coming together to form products, or they can be flipped and you can show products being broken down into back to their original reactants. Alright, so let's get into these now, the macromolecules of this cell. Again, we're going to be looking at carbohydrates, lipids, and proteins. Okay? Those pictures make me look hungry too. So first one we're going to touch on is carbohydrates. So carbohydrates are a class of molecules that range from small, basic simple sugar molecules to large polysaccharides such as starches. So again, starch being things like your breads and your pastas and your rice, simple sugar, table sugar, glucose, that type of thing. So the building blocks of carbohydrates are known as monosaccharides or simple sugars. They are monomers of carbohydrates. So basically, a monosaccharide is a very simple sugar molecule that is then brought together with other monosaccharides, and then they start to come together and start building more complex sugars or build into polysaccharides. So mono meaning one singular molecule. So for example, this molecule here, glucose, shown here in this form, and then glucose again in this other skeletal based structure here, just showing all the hydrogens and the oxygen bonding to the carbon skeleton here, or the carbon backbone rather, excuse me. So that would be a monosaccharide. But if you started to bring together multiple glucose molecules, or a glucose and then another type of monosaccharide of fructose, and they started to hook up or come together, they would start to form a polysaccharide. Okay? And these sugars or these carbohydrates are typically found in the molecular formula in some form of multiple of CH2O. Okay, so they're made up of carbon, hydrogen, and oxygen, again in a particularly fixed ratio. So if you were to, for example, look at glucose, glucose's basic molecular formula is C6, so 6 carbons, H12, 12 hydrogens, O6, 6 oxygen. So C6H12O6. Okay, so that's again a multiple. If you were to say multiply CH2O times a coefficient of 6, that's how you would get the formula or the breakdown of glucose. Okay, carbohydrates again are made of a carbon skeleton or backbone. Examples again real quick were glucose and fructose. So when we say skeleton or backbone, we're talking about the chain of carbons here. Other carbohydrates can have anywhere from 3 to 7 carbons as well. But the more common ones that we will see throughout lecture and throughout the readings and this particular course typically are going to be 6. And then monosaccharides like glucose, for example, are the main fuel for cellular work. So again, cells release energy from glucose when they break it down, and then an aqueous solution of glucose can also be injected directly into the bloodstream of the sick or injured to help revitalize and provide the energy needed for somebody. You see glucose listed, for example, in your Gatorade, for example, or your sports drinks as well, right? A glucose is, again, it's a simple sugar that can easily be shuttled in and out of cells, and that's what helps provide energy, either to supply energy to a part of the body, whether it's a muscle or a cell, or for glucose to be taken out of one part of the body and released and provide energy into another part of the body. Okay. So again, polysaccharides, as I mentioned, are basically simply taking the monosaccharides or the simple sugars, and they are starting to come together. Now, how do they come together? Is based off of dehydration reactions. Okay, this is a key term here, dehydration reaction, and it is exactly what you might think. Dehydration meaning loss of water, right? And we see that formation here. So, for example, to make maltose, which is a polysaccharide, okay, it's a more complex sugar-based molecule, it takes two glucose molecules to come together. So, we have glucose here, we have glucose here, our two reactants, and as you can see at their tail ends here, there is HO here and another H here coming off. That forms water, H2O. If we lose that H2O, then our product comes together and we're held together here by a common oxygen molecule. And this linkage is now maltose. Okay? So again, dehydration reaction. Okay? Another example here, glucose and fructose comes together to form sucrose. Again, you look at the ends here, the tail end of the glucose, the beginning of fructose, and you see that we're losing again an H2O molecule in order for these two monosaccharides to come together. So, simple terms, how does a polysaccharide form and how does it come together? Dehydration reaction. You have to lose a water molecule in order for two monosaccharides to link up. And then here are just some examples. So starch, storage polysaccharide in plants, long chains of glucose monomers typically. They may be branched or helical in shape. Animals store glucose in a polysaccharide known as glycogen. This is important, particularly for us as well as humans, because we do the same thing. We have in our muscle cells in particular, for example, we have glycogen. So, for example, for any of you guys that are hardcore athletes or workout, and I know a lot of you guys are in the armed forces, you are working out constantly, and then you are needing to eat and refuel. Well, a lot of times it not only is protein stressed in diets and exercise, but you need to have a good amount of carbohydrates or starches in your diet as well. And the reason for this is because the starches are going to be eventually come together in the form of glucose molecules binding together, and they're going to stay stored in your muscle cells, providing fullness to your muscles in the form of glycogen. Now, when you guys go out and you go work out again, or you're going on one of your missions or hikes that you guys do, for example, and you haven't eaten for a while, but you still need that energy to continue on and to push through your training, well, within your muscle cells, because of the carbohydrates you ate the hours or night before, that glycogen in your muscles will break down and release glucose monosaccharides, glucose molecules, into your bloodstream, and that's going to give you that energy that you're going to need to continue and finish through your training. So again, perfect example there. And then there's cellulose, which is the most abundant organic compound on Earth. It's a major component of tough walls that enclose plant cells. Okay, cellulose molecules are joined by hydrogen bonds. They form microfibrils or fibrils, Um, and again, so cellulose is seen and found only in plants. Humans, we do not have enzymes to digest cellulose, but it's still useful to our health. Think about that for a minute. Cellulose cannot be broken down, so when you eat a salad, for example, and you're eating lettuce, lettuce is a plant, lettuce contains cellulose. You're not able to break that down, but why then would lettuce be useful to us or healthy? Well, lettuce and other plant materials that humans consume for health reasons provides fiber, right? It's insoluble. We can't break it down, but it will help push things through, if you will, through your body. So, cellulose, also known as insoluble fiber, can be found in fresh fruit, vegetables, and other whole grains that might be rich in fiber or say that they're rich in fiber means that they contain cellulose. So if we can't break it down and we can't digest it, it's still going to help at least push everything else in and through our bodies. Okay, lipids, fats, okay, the tasty stuff, right? Everything that makes our food taste so good, but yet it can be so bad for us. Um, so with lipids, they're a diverse group of molecules that share a common trait. They do not mix well with water. This term is also known as hydrophobic. So again, something a key term you need to be familiar with. We will see and look at three important types of fats or lipids. So fats is a type of lipid. Phospholipids is another one. Phospholipids are lipid molecules that make up the cell, for example, or make up the membranes of your cells, and steroids. And yes, I am talking about both naturally occurring steroids as well as the anabolic type of steroids. These are forms of lipids. Okay? And so as you can see here from the pictures, we're looking at an example. So again, oil. Oils, different types of oils are considered lipids. They're hydrophobic, and obviously they are not mixing well with water. Here is kind of a up close view of what your cell membrane, for example, looks like. So on one end you have water, or you would have the cytoplasm, the aqueous solutions of the cell. That is followed by a polar head group, which is hydrophilic, meaning it does like water and it can handle water. So the hydrophilic heads or polar heads stick out into the water or aqueous solutions. And then you have the hydrophobic or non-polar tails. And these are the ones that are made up of lipid chains that we'll see in what they look like in a minute. And they don't mix well with water, but they get along great with each other. So hydrophobic molecules will come together and they will avoid water. And this is exactly how your cell membrane is composed of and looks like. So fats, one of the forms of lipids, is a large lipid made up of two kinds of smaller molecules, a glycerol and fatty acid. So the fatty acids are these chains here of carbons, okay? And then the glycerol group is again made up of typically, a hydrogen and oxygen combination. So glycerol consists of three carbons, each bearing a hydroxyl group. So again, you have carbons and oxygen and hydrogens up here as the head group here for the glycerol. And then the fatty acids, or the hydrophobic chains, are going to consist more of carboxyl groups and hydrocarbon chains. Okay, so they're mostly hydrogen and carbon. No oxygen is found in the chains, and so that's what makes it hydrophobic. So the chains here do not like water because there's also no oxygen as well. Okay, whereas the head groups or the glycerol, that part, for example, could interact with water because there is a hydroxyl group, so oxygen, hydrogen, and a carboxyl group. And then, of course, you've heard the term saturated fats, unsaturated fats. One is considered good, one is considered bad, but why? Well, here is why. So, saturated fats or saturated fatty acid are basically straight chains. There are no double bonds that you see here in the hydrocarbons, and therefore saturated fats or products that say that they're saturated in fat are going to be thick and solid at room temperature. Whereas you have unsaturated fatty acids, or a product that tells you that there is unsaturated fat, means it does contain double bonds in the fatty acid chain. This causes kinks to occur, which makes it more flexible. So oil, for example, certain oils are considered unsaturated because they're liquid at room temperature. And the reason that they are able to be liquid at room temperature is again because of their flexibility, because they have these kinks in their fatty acid or hydrocarbon chains. Okay, so that's the difference and why saturated fats are considered bad compared to unsaturated fatty acids. Trans fat then is something that we've also heard about lately. So trans fats are, and again, there's a small portion in your book that you should definitely familiarize yourself with. But basically, new research has shown that trans fats are produced in the process of hydrogenation. Okay, and this poses a greater health risk than, say, saturated fats. Now, you do have um, naturally occurring, trans fats, and these are found in very small, minute, ratios. So, uh, for example, milk, meat products will from straight from animals do contains traces of trans fat, but nothing that's going to pose a great health risk. But products that are processed in factories, so your junk foods, your your frozen foods, your frozen pizzas, some chips, things like that, those may contain trans fats, and those are created in an industrial process that basically pumps and adds hydrogen to liquids, oils, for example, and that's going to make them more solid. So, by increasing the number of hydrogens, you're going to basically ultimately cause the fats to become more saturated and become the chains to become more straight. Okay, so again, making oils more solid. And again, this can obviously lead to various health problems down the road. Okay, so that's trans fats. Okay, trans fats again, they raise your bad cholesterol or LDL, your low density lipoproteins. We'll actually talk about cholesterol a little bit later on as we progress, probably in the next lecture next week. But for now, just understand that trans fats raise your bad cholesterol, which is LDL, and lower your good cholesterol, the HDL, the high density lipoprotein. That's what the HDL stands for. And then again, food makers use artificial trans fats to enhance the flavor of food, the texture, and of course the shelf life of processed foods. So even though it might not say trans fat on your food labels, you might look and you might see something that says partially hydrogenated oils. And again, they're artificially in the in the factories pumping hydrogen into your foods. And right here is a perfect example where you have vegetable oil, which is liquid, typically at room temperature, for example, but as the process, as you increase the number of hydrogens, it becomes less flexible and becomes more and more solid. Okay, phospholipids, as I mentioned, are major components of your cell membrane. So, phospholipids typically contain two fatty acid chains attached to a glycerol. So, remember, the glycerol is the polar head group, which is hydrophilic and can handle and likes to be and interact with water. But then you have the carbon and hydrogen chain here, the fatty acid chain, which is hydrophobic. And they will cluster together because hydrophobic and hydrophobic like to stick together. Hydrophilic will likes to interact with the water. And again, your cells, there is water and aqueous solutions found on the outside of your cell, and within your cell, there's an aqueous component known as the cytoplasm. Okay? So again, this would be if you took a cell and you split it in half, what you would see. So inside you would have the cytoplasm or water. On the outside of the cell would be water as well. So again, in the red here you have your polar head groups, which are hydrophilic, and then in the middle here in the yellow portion with the red tails is your lipids or your phospholipids, the fatty acid chains, which again do not like water. They are hydrophobic. Okay, steroids, the other form of fats. Okay, so we've all heard about steroids. We all know about steroids. They're in the news constantly, particularly amongst athletes and sports. But steroids are also naturally occurring. You do have steroids that your body makes naturally. Um, so, steroids, background obviously, are lipids in which the carbon skeleton contains four fused rings. Okay, so with all the other lipids and the fats and the saturated fats and those kind of things that we were looking at previously, we saw that they were made up of also carbon skeletons, carbons and hydrogens, but they were in chains. Steroids, as you can see here from the examples, are made up of rings. So each point here in a given ring are carbons. So there's still carbons, obviously, that make up these rings, but instead of chains, they're rings. Cholesterol is a common component in animal cell membranes and is also a precursor for making steroids, including the sex hormones that we are or should be familiar with. So, for example, with males, it's testosterone. With females, it's estrogen. These are steroids, or these are steroid-based hormones that um, contain and have cholesterol. Um, and then you do have anabolic steroids as well. So anabolic are the ones that people will inject into their bodies, into their muscles directly, to again get that enhancement, that increased muscle mass or bone density comes from the anabolic steroids, which again are usually synthetic variants of testosterone, and that's because testosterone, the male hormone, causes again the buildup of muscle and bone mass. Um, but obviously, steroids, even the anabolic types, can be used um, for medical purposes, for medical reasons, to help treat diseases. So, muscle wasting diseases, for example, um, some kinds of cancer, um, autoimmune diseases, um, such as lupus or multiple sclerosis, arthritis, asthma. These can actually be treated with steroids. Uh, so not all steroids that are synthetically made are used only to build muscle and to create juice heads, if you will, or, you know, bodybuilders or people who just want that aesthetic look. Okay. And then, of course, I've posed here just some from this graphic here, the potential negative side effects of the use of anabolic or synthetic steroids. Uh, so obviously, both men and women will use and do tend to experiment and try steroids at some form or another. There are oral-based steroids, so pills that you can take, but typically you have to take a large amount of the pill form of steroids to get the effects, whereas obviously the injectable types are much quicker and to the point, if you will, in getting the results that you may be looking for. Now, again, potential side effects amongst men, for example, is the development of breast tissue. Um, Baldness can occur as well, so you will lose hair on the top of your head, but there are cases where you will gain hair in other places of your body. You might get increased chest hair, back hair as well. Can cause enlarged prostate and shrinkage of testicles, which is very common and what you hear a lot of times. Um, Now, depending on how long a male uses these anabolic steroids, um, the size of their testicles can and will usually come back to normal if they cycle off or stop taking steroids after a while. But prolonged use of steroids uh, can be detrimental and have these negative effects. And then again, amongst both men and women, you do have the bloating, nausea at times, impotence. So women can lose the ability to get pregnant. Men, you reduce your sperm count. Aching joints, aggressive behavior, mood swings. Honestly, that's kind of up in the air. I'll tell you from both personal as well as professional experience uh, with steroids and with people who have used steroids. Um, you know, some people might say that roid rage exists. Others might say it's dependent on just who you are as a person normally. So normally, if you are um, an aggressive person, then yes, maybe steroids might help enhance that aggression. But if you're normally a calm person, it's not going to cause you to flip automatically or that quickly to becoming a more aggressive person. So, just various things to keep in mind when you hear about steroids, particularly in the media. Okay. Um, so again, different types of steroids that occur and that are there. So you do have things like sex hormones, which influence the sex difference and support reproduction. So we're talking about estrogen, uh, progestogen is another one, androgens as well. Then you have corticosteroids, which are produced naturally in the adrenal cortex vertebrae. And there are some natural products there called glucocorticoids that regulate metabolism and immune function. And then you have things like mineral corticoids that help maintain blood volume and control the renal excretion of electrolytes, excuse me. And then again, as we touched on, we do have then anabolic steroids. And again, there are both natural anabolic steroids as well as the synthetic types as well. And the anabolic steroids interact with androgen receptors, and this is going to trigger and cause an increase in muscle mass and bone mass and density as well. So those are steroids and background on steroids. Okay. And then lastly, our other final major macromolecule here are proteins. So proteins are a polymer of the small building blocks called amino acids. So I provided you with a video, a YouTube link here that I definitely want you to be familiar with. And it discusses and does a great job of animating the process of protein synthesis. That is bringing the building blocks or the amino acids together, and they come together, they form bonds, and they create a chain. And basically, a large chain of amino acids make up a protein. Okay? Proteins are found throughout the body and play important roles in the cell in particular. And again, as we get more and more into the cellular biology, I believe over the next week or two, we are going to see proteins that act as enzymes. We're going to look at transport proteins, so proteins that help, for example, form tunnels within the cell membrane to allow molecules and other important nutrients to go in and out of the cell. Defensive proteins, so proteins involved with the immune system. Signal proteins, muscle cells, obviously, and structural proteins. Those are all made up of a string of amino acids coming together. And what you'll also see is that the shape of a protein determines function. That's a key thing amongst molecular and cellular biology overall. It's a central theme that, structure determines function. And that's most important amongst proteins. So again, please be sure to review that video on that YouTube link there for protein synthesis. Okay, and then what I've done here is I've simply just illustrated. This is what's known as the central dogma of molecular and cellular biology. Even if you are to study genetics, you'll hear the same thing also reinforced. So DNA gets transcribed into messenger RNA, and that messenger RNA contains the code needed to be translated into ultimately protein. So all the proteins that make up who you are is determined ultimately and is encoded in your DNA. So to go from DNA to protein is ultimately what's known as the central dogma of molecular biology. Okay, so the dependence of protein, again, function on a protein's shape becomes clear when a protein is altered. Okay, so there can be times where whether it's naturally occurring or there is damage to your DNA, for example, through mutations, this can cause proteins to denature. So when a protein unravels, it loses its shape and ultimately its function. And we will see some examples of this later on. But, for example, cancer is a big major disease that can be triggered by the denaturation of protein. So there are some proteins out there that are needed to be in a certain shape and that function to help prevent, say, cells from growing out of control. But if the DNA that encodes for that protein is mutated, or that protein gets denatured somehow, and it loses the ability to slow down or stop the cells from multiplying, then cells will uncontrollably multiply and constantly regenerate. And the definition of cancer is the uncontrolled, multiplication or generation of cells. So again, proteins, again, shape determining function. Okay. Some examples as well, along with cancer, of protein denaturation and their results are Alzheimer's disease, Parkinson's, and diabetes as well. Okay, so the amino acids. Okay, so again, these are the building blocks of proteins, and they are 20 to some cases 21 essential amino acids. And what I want you guys to know from this chart and from this particular graphic here is I want you to know their single letter codes and their properties. So what do I mean by that? So for example, I want you to be able to identify the polar uncharged side chains here. So the polar amino acids, for example, are serine, threonine, asparagine, and glutamine. And I want you to know their single letter codes. So serine is S, threonine is T, asparagine is N, glutamine is Q. So I want you to know that. I want you to know, which ones are, negatively charged, for example. So aspartic acid, D, glutamic acid, E. Um, Down here, this larger group here, for example, these are the hydrophobic amino acids. So these are the amino acids that do not like to interact with water. They will cluster together, and they are alanine, isoleucine, leucine, methionine, phenylalanine, tryptophan, tyrosine, valine. Okay. Now, the reason I want you to know the single letter codes is because that's how you will see them in your research and how you will see them when you read through scientific papers. A lot of times they're not going to spell out the protein or the amino acids that make up a protein in its entirety. A lot of times you will see a string of single letter codes, and you're going to need to understand what those single letter codes mean and what type of protein is that ultimately forming. So if you see a protein listed in your paper and it contains a lot of isoleucines, leucines, phenylalanines, and tryptophans, and very few serines and threonines, you're going to know, for example, based off the fact that majority of those codes are hydrophobic, that chances are that protein is not going to be found in the cytoplasm or found in the bloodstream or in water, but maybe it's going to be embedded in the cell membrane because it has so many hydrophobic amino acids. Okay, so that's what I want you to do there. So again, know the 20 to 21 essential amino acids as stated here on this graphic, and know their properties. So know which class they belong to and their single letter codes. Just a quick little fun mnemonic to kind of help jumpstart it and help the process. So this is one that I came up with a while back, but for the polar amino acids, think about this. Okay, so the single letter code is STNQ. You could probably spell that out as stink. Well, guess what? Polar bears stink, right? So that's just one. So polar meaning the polar side chains here, stink, serine, threonine, asparagine, glutamine. Okay, so that's one way to remember that these four make up the polar amino acids. For hydrophobic, this one's a little bit more out there of a device that I came up with. But basically, if you were to rearrange some of the single letter codes here, what you'll come up with and what I came up with is my vile wife. So, my vile wife. Now, obviously, I'm not married, but basically what that means is I'm thinking about the word vile as in hateful, spiteful, right? And these are hydrophobic, or they hate water. And the reason I came up with my vile wife is because methionine and tyrosine make up my, my. Methionine is the M. Vile is from valine, isoleucine, alanine, and leucine, VIAL. And then wife comes from tryptophan, tyrosine, and phenylalanine. So you spell wife WYF in this sense. So M is the my part, vial, valine, isoleucine, alanine, leucine, and wife, tryptophan, tyrosine, phenylalanine. My vile wife. She's hateful, she's spiteful, she doesn't like water. She's hydrophobic. Okay? So again, just a fun little mnemonic device there to kind of help you group and memorize the amino acids. So again, work on that. And then how do they come together? So uh, again, amino acids will come together, um, and they also form in a dehydration-like reaction. And this is called a peptide bond. So when amino acids come together to form proteins, that is known as a peptide bond. Okay, the R groups, or the side chains of proteins, is what gives proteins their characteristics. So again, hydrophobic amino acids may cluster together to form a globular protein. Hydrophilic amino acids face the outside, helping proteins to dissolve in aqueous solutions. And then hydrogen bonds and ionic bonds also determine the shape of a protein, as do covalent bonds like disulfide bridges. So again, different types of bonds help determine the shape of a protein, and remember, shape ultimately determines function of a protein. Okay. Um, then these were just kind of again touching on diet essentials or diet basics. So again, with regards to your diet and your nutrition value of these macromolecules. So fats provide a highly concentrated form of energy. It enables your body to transport, store, and absorb vitamins like A, D, E, and K. They provide insulation and protective cover of your vital organs. Carbohydrates provide energy as well. They're obviously not in high concentration as fat is. And the energy comes in the form of the food that we eat, such as the grains, breads, and pasta. They are converted into glucose that the body uses either right away or stores later. So when you hear marathon runners carbo loading the night before a race, right, they're eating a lot of bread, they're typically eating a lot of pasta. This is to increase the amount of carbohydrates in their blood, which then is going to be stored in their muscle cells as glycogen. And then once they go out and they race the next day, that glycogen is eventually going to break down and provide glucose, the energy necessary for them to continue on with their race. Protein is also stored mostly in the body as muscle. It generally accounts for 40 to 45% of our body's total pool, and it helps form the structural component of body tissues and is used within many biological processes. Proteins are used to make up enzymes, antibodies to help fight infection, as well as DNA, the building block of life. Okay, and again, we're gonna be diving into many of these aspects in greater detail as we progress. Okay, so that is lecture 2. That's where we're at, and that's macromolecules. Again, if you have any questions, comments, or concerns, please feel free to contact me as soon as possible through email. Make sure you look over the slides, read through the chapter, re-listen and to this particular recording over and over again, and make sure to do the assignment as well. Okay? Thanks very much.